Thanks, Rui, for this introduction. Also, thanks for both Rui and Caroline for inviting me to speak. So here's going to be my plan. So I'll just give you a very brief introduction to the multi-species coalescent. This has many applications, but today I'll mostly focus on the use of the multi-species coalescent to infer species trees. So I have an example that's based on the African mosquitoes, Anopheles gambia species complex. If I don't go over time, normally I do, I'll just show you a few slides about inference of cross-species gene flow. This is something that we have been working on in the past two or 3 years. So we more or less all know the standard coalescent process. So here's just a really quick diagram. On the left is the more traditional Fisher-Wright model, describing the process of reproduction and random sampling when the gametes are used to generate targets for the next generation. So it's a binomial sampling model. In the literature, it's often written as the Wright-Fisher model, but I think it was Anthony Edgeworth in Cambridge who argues that who pointed out that Fisher's paper was 1930, Wright's was 31, so it should be called Fisher model. So just take the middle ground, we just call this the Fisher-Wright model. So the sample version just assumes the constant population size, non-overlapping generations, random mating. So this is like a generation by generation. So the coalescent was developed in the 1980s. So it's the same process, but viewed backwards. And also, instead of focusing on the whole population, so this is just for illustration, this is a small population of only 10 individuals. So we are deployed, so there are 2n or 20 sequences at any genomic segment or locus. So when you take the coalescent view, you run time backwards. So instead of focusing on the whole population, you focus on the sample. So here we have a sample of 5 sequences. So you focus on the sample and then think about the genealogical tree describing the sample. So then you have this exponential waiting times. So the coalescent initially is really fast and then gets slower and slower. So the rate of the coalescent is given by 1/2n. So that's the rate of coalescent in one generation. And then for every pair, and then the rate gets lower and lower when you have fewer pairs to correlate. So I think it's important to recognize that both models are describing the same biological process. It's that this coalescent view is extremely useful in molecular population genetics or population genomics today in analysis of sequence data, genomic sequence data. So the multi-species coalescent is just a very straightforward extension of that single population coalescent to the case of multiple species. So here in my diagram, I have 2 populations that are called A and B. So there's a population divergence time or species divergence time. So you might talk species and populations are synonymous. So then we have 3 population sizes instead of just 1. there's also a species divergence time. So there are 4 parameters instead of 1 parameter as in the single population version. So usually this model is aimed at analysis of molecular data. So we scale time by mutations. So basically we multiply those things, these parameters. I just want to confirm that you can see my mouse pointer. Is that correct? Okay, yeah. So, so we multiply the divergence time by the mutation rate, which we call tau. That's still called the species divergence time, but it's the time t in generations multiplied by the mutation rate in generations. That gives us the genetic distance. So a tau of 0.02 essentially means that from this time to the present, you see a sequence change of 2%, or 0.01, that's 1%. Yeah. So theta is 4 mu, that's the like really fundamental population genetics parameter. It's measured in the same way. So theta for the human is slightly less than 0.1%. So it's basically heterozygosity in the population. So so that's the extension. So I just have a slide about some features I want to describe. So in terms of parameters, we have 2 sets of parameters, divergence times and population sizes, instead of just 1 population size for a single population. Another point is that lineages or sequences join independently in different populations. So here it's got an example of 3 species. Those are the parameters. 1, 2 divergence times, 5 theta parameters or population size parameters. So when we trace the history of the sequences, they join in the different populations independently. So in a way, you have a coalescent process in population A, big A, from time 0 to tau AB. So in this time interval, you have the single population coalescent really happening independently. So that concerns sequences A1, A2 here. But beyond this point, you got a different population that's called AB. So this common ancestor A and B. So coalescence happens here in this population also independently. In my particular example, I have 1 coalescent in population B, and then this 1 sequence goes into AB. No coalescent in A, so both go into AB. So initially 3 sequences go into AB, so they will coalesce. There's 1 coalescent event, and so on. So lineages join independently in different populations. A coalescent reaches reset when lineages enter a new species. So that means that inside population A, the rate is given by the population size of A. But as soon as you enter population AB, you have a different population size, so the rate is different again. So it's all very intuitive and straightforward. Another point is that genes split before species. This is pretty much obvious, which basically means that in this diagram, a sequence from A, let's say A1, and a sequence from B, let's say B1. So A1 and B1 will have a coalescence time. In this diagram, it actually goes to all the way to load 4. That's the root. So anyway, so the sequences from 2 species must have a coalescence time that's older. So before and after, like confusing terms here. So maybe I should just say older. So the T, small t is our coalescence time. T A1, B1, or TAB, just like the coalescence time between 2 species must be greater than the tau AB, the species divergence time. You could also say that gene trees must fit inside the species tree. So this kind of this looks like a pair of trousers, but with 3 legs. So this kind of diagram is very common in this area. Some of the reviewers force us to use a diagram like this. Sometimes we had a simple species tree, they were not happy. Just could you redraw the species tree, in this way so that looks nicer. So you could say that gene trees must fit inside the species tree. This actually causes a lot of trouble for us when we implement the model in the MCMC Markov chain multicolor algorithm. So also what you just mentioned, the terminology. I think the earliest term used by Professor Takahata was interspecific coalescent. This is a coalescent running between species. And when Bruce and I had our paper describing the gene tree densities and so on, I'll come back to that a bit later. We use the word censored coalescent. This is because if we look at the coalescent for population A, A1, A2, they have not coalesced when we reach tau AB, but we have to stop the coalescent process. So that's what we mean by censored. In a single population coalescent, you have to wait until all the sequences coalesce. Here you have 2 lineages, but you stop the process. But I think nowadays this term multi-species coalescent has stuck. Everybody is using this term now. So we are just using this as well. So here's a bit of history. Maybe I'll just very briefly go through this. There's a paper by John Gillespie and Chuck Longley. So this is something I didn't see really early on. This is even before the coalescent papers. So they were dealing with something about the molecular clock and so on, but they mentioned that for 2 species, the difference between the 2 in terms of number of substitutions is the sum of a Poisson and a geometric random variable. So they really clearly recognize the 2 components of the sequence divergence between 2 species. You have this part that's the species divergence. This part small t, that's the coalescent in the ancestor. And then it turns out when you talk about the mutations, number of mutations, this is a Poisson, this is a geometric. Also, Takahata in 1986 had a paper about estimating, using exactly this same model, 2 species. He was dealing with the estimation of the ancestral population size for the human chimp common ancestor. I remember I was very suspicious. I just thought this just asking too much, Human chimp common ancestor, how could you estimate the size of the population for the common ancestor? I guess nowadays I just spending most of my time doing this. But I didn't recognize, the idea was fine, just thought it was putting not much information for doing that. Anyway, it's really a paper that's ahead of its time. In the case of 3 species, the earliest paper is probably Dick Hudson. This gave the gene tree, species tree mismatch probability for 3 species. So I have a slide later about this. And Takahata and Wenxiang Li, so they are working on these models. Also, there's a branch of coalescent theory dealing with population structure. So the people here developed the so-called structured coalescent, which is useful for us in models like the IM, oscillation, migration models. We can use the structured coalescent models. So here, this is what I found to be the earliest 3 legs trousers that are published in the literature. And as I say, this is like a standard way of presenting gene trees inside the species tree. So the multi-species coalescent has many applications and extensions. So you can use that to estimate the species divergence times and population sizes. You could also estimate migration rates or integration probability. So we have programs like RM, and you can also estimate, infer species boundaries or do so-called species delimitation. This is like a controversial area that I won't discuss today. So today I'm mostly focusing on the species tree estimation. So one review paper that's very nicely written by James Digley makes the point that the multi-species coalescent is the basic null model. And on top of that, you can add other factors such as population structure, hybridization, recompilation, and so on. He also was not happy with some of the other papers trying to distinguish hybridization from called incomplete lineage sorting, something like that. He said these descriptions are unfortunate. Yeah, I entirely agree with him. As I said, the coalescent is basically the biological process of reproduction viewed backwards in time. So if you are trying to distinguish coalescent from hybridization, you are always saying that I want to distinguish between species that reproduce and species that hybridize. I think, the fact is that all species reproduce, but some also hybridize with other species. So yeah, I think this is a very nice review paper. You can take a look. So now I'm going to give you a bit more introduction of the MSC. So they have mainly two lines of research in a way, developing the theory. One gives you the probability distribution of the gene tree topology. So my big G here refers to the gene tree topology. And then my small t here, that represents the coalescent times or branch lengths on the gene tree. So one line of research develops this gene tree probability. So there are probably certainly more than 10, but perhaps about 20 papers in this area. Another line of research is to give you the joint probability distribution of the topology and also the times. So this turns out to be actually simpler. So we had a paper that's about 20 years ago. F of G, you can perhaps argue that this underlines all the summary methods. So summary methods of inference, they use phylogenetic methods or programs like RAXML to infer the gene trees, and then use the gene trees as data to do all sorts of things like inferring species trees or inferring integration. So then obviously, given that you treat the gene tree topologies as data, you need to know their probability distributions. So that's why F of G is important. This term, that's the joint density, is useful for so-called full likelihood methods. This would include maximum likelihood Bayesian methods. Typically, these are applied to the sequence alignments directly. And you want to calculate the probability of the sequence alignments. That's called the phylogenetic likelihood. And so you need branch lengths or coalescence time. And you need the joint density of the topology at the time. So this term is useful for that. So I'm going to give you a bit of flavor of both. So here, this is the 3 species case, 3 species, 3 sequences. So this is actually in the paper by Hudson in 1983. It's a fairly simple argument. So if this is the true species tree, so example here uses human chimp gorilla. In the earlier days, I think before 2000, human chimp gorilla was used a lot in these kind of discussions. So given the 3 species tree, we talk about the possible gene trees for the 3 species. You get 1 sequence from each. So this is one gene tree that is formed by having a coalescent in this time interval between the 2 speciation times. So if you have a coalescent here, your gene tree will match the species tree. Otherwise, all the 3 sequences will go into the common ancestor HCG, and then the 2 coalescent events will happen at random. So then those three possible trees here will just be equally probable. So in a way, we have like a 4 gene trees, but these 2 are different from the species tree. They have different topology, and these 2 have the same topology. So the so-called mismatch probability is the sum of the last 2. And that's basically the probability that there's no coalescent in this interval. And then multiplied by 2/3. If there's no coalescent, you got those 3, and the 2 of them give you the mismatch. So you got 2/3. This term is from the Poisson variable, Poisson event. The probability of observing no event is exponential minus lambda t. Lambda is the rate of the Poisson process. That's 2/theta. T is the interval. That's this interval. Or you could also get this from the exponential distribution of the coalescent waiting time here. So if the waiting time is exponential with the average to be half theta, then the probability that you don't have an event or the exponential waiting time is larger than this interval is given by me. So I'm just giving you too much of the details here. So it's just like the probability of no event in the interval multiplied by 2/3 gave you the mismatch probability. For the human chimp gorilla, the mismatch is fairly stable, about 30% from several genomic papers. It's 30%. So that means in 70% of the genome or genomic regions, the true genealogical tree matches the species tree. But in the rest, the 30%, there's a mismatch. Okay, so that's the probability of the gene tree topology. Now, this is the joint density of the topology, big G, and the coalescent waiting times. So we have this example here. So the samples include 2 A's, 2 B's, and 1 C. So it's essentially the similar kind of exponential waiting times, exponential distribution densities multiplied together because the different populations are independent. You just need to allow for the probability that the coalescent process is truncated or censored in the sense that Bruce and I talked about before. So that there's a possibility that 2 sequences or more than 1 sequence can leave the population and enter the ancestor. So the process is censored. I think one of the terms here, this one. This is the lot exponential density. It is instead the probability that there's no coalescent in this time interval tau A when the rate is 2 over theta A. So you have to accommodate that. But anyway, it's very straightforward to write down. Although when the tree is big, you just have too many possible gene trees. Okay, so that gave you a flavor. My work, this talk, both mostly based on full likelihood method. So I just gave you a flavor of how that's done. If we do max likelihood, we will be, let's say we talk about max likelihood method for estimating species tree. So the main thing is that you have to average over all the possible gene trees, both topology and times, because the gene trees are not observed. So you get this likelihood term, that's the probability of the alignments for the different loci that's represented by I, given the species tree, and the theta will be, big theta include these two sets of parameters. So there are mainly two terms, two components in the model construction. This term I already mentioned in the previous slide. That's the joint density of the gene tree and the times given the species tree of the parameters. This term, that's the probability of the sequence alignment at locus I given the gene tree topology branchness. Some of you may recognize this is the so-called phylogenetic likelihood. That's based on Joe Felsenstein's paper of 73 and 81. So this is essentially in all the likelihood of Bayesian programs that you know about. So the difference from the traditional phylogenetic programs is that you got this term here, and also you have to sum over all the gene trees and integrate over the coalescence times. So this sum and integral, that's the trouble because the space is huge. So if you do max likelihood, you won't go far beyond 3 sequences. So we have a program for doing 3 sequences that's called 3S because we have no ambition of moving to 4. So we are just doing the integral numerically and summing over all the gene trees by enumeration. The Bayesian version is more manageable. So basically you build a Markov chain Monte Carlo sampling both the species trees and parameters as well as the gene trees at times. So the gene trees are sampled. They are part of the state of the Markov chain Monte Carlo. So you do this through so-called MCMC marginalization. So just like a lazy, expensive way of doing integrals, really. Okay, so the theory is straightforward. There's nothing new here. Just you could get this idea from any statistical textbooks, as long as the formulation is clear. It's just the calculation is difficult. So here, just to elaborate a bit more on that, there are 2 components. So the MSC density of the gene tree and branchness, that's straightforward to calculate. The phylogenetic likelihood is also straightforward to calculate. You normally do some reduced Cantor model or HKY if you like. That basically relates the tree to the sequence alignment. And then, yeah, everything is straightforward except that it's expensive. So here, just a sketch of the MCMC algorithm. So our program, BPP, pretty much is doing something like this. So we just initialize everything, species tree, parameters, gene trees. You have a move to change the parameters, have a move to change the gene trees, topologies at times. You can also have a move to change the species tree, and then just save once in a while. So the idea is that the frequency by which you visit the different species trees, they will be the posterior probabilities. So so we have been struggling with this for about 20 years. I think the fundamental difficulty, there are lots of challenges, but the biggest is probably just this constraint that I mentioned earlier, which is that the gene trees should fit inside the species tree. This causes trouble. So the gene trees are species trees, they constrain each other. So we have been mostly just spending our time to design algorithms that somehow accommodate that. So the first one, for example, is like a change, a move, MCMC move that changes the species divergence time. That's called tau. So maybe I'll go to the slide here. So this is the species tree. Let's say we are changing this tau AB right now. The thing is that there are lots of gene trees. If you have 1,000 loci, there are 1,000 gene trees. And then we say that the sequence divergence time between A and B, let's say A1, B1, that ages on this tree is here, that has to be older or larger than tau AB. So the smallest in this case is somewhere here. So A2 and B1 have the divergence time that's here right between the two taus. So if you have 1,000 loci or gene trees and you scan all the gene trees found the smallest sequence divergence time, it's very much very close to tau AB. And then if you are going to change tau AB when the gene trees are fixed, you are constrained very tightly, so you can't really move up because all of them are, have to be older or larger than tau AB. So what we did was to do this so-called rubber bound algorithm. So we are saying that we have a piece of rubber band. So they are fixed at the two ends, and then we hold the rubber band from this point representing tau AB, and then pull up or down. And then there's coalescent times called points ABC here. So they just move together. So we just use the analogy for the the movement of the points on the rubber band, and this worked very well, we found. So yeah, so it's the things like this that just basically occupy our time. When we move between different species trees, the problem is much harder. So this the change for tau, that's like a update of a parameter in the model, that's simpler than MCMC algorithms which move from one model to another. So species trees, different species trees are clearly different statistical models. So we need to do this kind of trans-model MCMC. It's quite hard. So I'm just going to show you one diagram that's what we did using the nearest neighbor interchange, NNI. So this was done in 1914. So suppose the current tree is like this. So we cut this branch, reattach here, you get a different tree topology. So this is called NNI, applied on rooted trees. Again, if you want to keep the gene trees unchanged, you can't do this, because when you change the species tree, all the gene trees become meaningless. So what we did is to change the gene trees together. So we cut off the species tree from here, and then there's all gene tree branches. They are cut, pruned off, and reattached here. I remember this was quite a lot of effort. Before Bruce and I did the work, we were not sure whether this would work. There were some issues which caused problems, and then we were ready to abandon the idea that it was part of the problem. realized later. Later on, we developed some other algorithms such as subtree SPR, subtree pruning regrafting. Even though SPR is a bigger move than ANI, that was much easier because we were really certain that the idea would work. I just realized that when you know the idea must work, it's actually easier to do the work. But if you're always thinking that the idea is probably lousy and doesn't work, you really just give up. It's actually harder somehow. So I'm going to show you a few slides just contrasting these full likelihood methods with the summary methods or two-step methods. This one, we had a paper that's just accepted of advanced publication here. This was probably like 7 or 8 years in the making, quite a lot of effort. So we were actually doing max likelihood in this case. We are dealing with exactly the case of 3 species, 3 sequences per locus. So here it's just a simple simulation or calculation that contrasts 3 different methods. Max likelihood, as I explained in the previous slide, using the program 3S. Two-step, that's equivalent to the method known as MT-East. This is done by Liang, Liu, and Scott Edwards and others. And there's another one that we call the independent sites ML. This is similar to the SVD quartets, except that we are applying this to rooted trees and 3 species. So here we have M loci, N sites in the data, but we are fixing the product, which is the total number of sites fixed. So if you do maximum likelihood, you get an error rate like this. So here on this N, that's just using 1 locus with 10,000 sites. Here on the left, you have 10,000 independent sites. So the big problem with these 2 ends is that you have some identifiability issues. In the middle, you see the errors are not so different. So these summary methods, they do have identifiability issues because they use summaries of the original data. I think it's probably in one of those earlier papers, somebody said that independent sites are more informative. We just point out that's not correct because you're not using the information correctly. If you do 2 loci of 5,000 sites will be more informative than 10,000 independent sites. So the error here is much lower than here. So here's another slide with some earlier simulation studies. I guess there were not entirely simulation studies, but they compared different methods. So BPP compare with Astro and SVD quartets. So this is the percentage of getting the right tree. So the trees here, they are difficult. And the full likelihood methods are much more efficient. They are not really comparable here. This is another paper by Kim and Dinkler. So I'll just very quickly show you some real data analysis. We apply this to the genomic data for Anopheles gambia species group. The data were mostly from this paper in Science, but we did a bit of modifications. The main message in the paper is that the X chromosome gives a tree that's totally different from the autosomes. So here are the sliding summaries of sliding windows analysis in the genomic paper. We support that main conclusion. Our analysis here is using BPP running across the blocks of 100 loci. Each bar represents one block of 100 loci. So these colors represent different species trees in a way. So the X chromosome has very different trees from autosomes. So we support their conclusion that way. So we also had arguments. We agreed with their arguments. We have stronger evidence that X chromosome tree is actually the truer tree, and autosomes are the result of integration. But our trees are different from theirs, and we argue that their trees are wrong, ours are right. So this is our tree. There's an integration this way. The last thing is that when you add the well-known integration starting from this X chromosomal tree, you do get the autosomal tree. This is not the case, not so simple with their trees. So here's a summary of the difference between the two. So I guess we agree with some of the conclusions they drew, but disagreed on the details. We argue that their trees were wrong in the right way, in the way that we expect, because they use sliding windows. That's like a concatenation, which is meant to give you wrong trees in particular ways. So I still have a few minutes. I'll talk about some work we did recently on inference of gene flow on species trees using genomic data. So this is a paper that's done by my postdoc, Ziheng Yang and Thomas. So we are looking at the impact of gene flow on species trees. The idea is that when you have gene flow, you could end up with a wrong species tree. So actually, we apply this like more theoretical study to the Allophilus example. We confirm that using the estimates from the data, we confirm that starting from the X chromosomal tree, you do end up with the autosomal tree. So very briefly, I'll just go through a few slides about the recent work implementing multispecies coalescent with integration. So currently in the program BPP, we have these different types of integration models. So there are these kind of, sometimes you get a hybrid species with two parents. That's the difference from the traditional species trees. So there's 5 parameters. They represent the probabilities of integration, so like the intensity of integration. In the literature, you must see the symbol gamma being used. That's exactly the same thing. So in our program, there's too many gamma inverse gamma distributions. We just found it so confusing. We found another symbol that looks a little bit similar to gamma, but it means the same thing. Yes, so this is the same. So we analyzed the Allophilus data to estimate the integration probabilities. So here's the A to GC integration. This is a very well-known one. There's another one from R to Q that's plotted in those two. So the integration probability tends to vary among the genomic chromosomal regions. Okay, so the another interesting study we did was to look at the impact of gene flow. So this is a really exciting case. It's a little bit complex. I'm going to take time to explain. So suppose we have two species, but there's a little bit of gene flow going from B to A. Then, even if it's a little bit of gene flow, you may end up with a situation where the within species difference is actually apparently larger than between species. Or more precisely, we take two sequences, A1, A2, and one sequence B. So I got two measures of closeness. One is just think about these two possible gene tree topologies. G1 has A1, A2 together. G2 has A1, B together, or A2, B together. So, so you can have a bit of gene flow between the two species so that G1 is less probable than G2. Okay. Another measure we use just the average sequence distance or average coalescence time within species or between A1, A2, or between A1 and B. You could have a situation where the TAA has expectation that's larger than expectation of TAB. So this I find very exciting. So you get a little bit of gene flow, fairly realistic, and then we basically characterize the parameter space to say that when this M, that's big N, small m, is measured by the expected number of migrants per generation. When M is larger than about 0.5 here, G2 has a higher probability than G1. Or similarly, if M is a little bit bigger than something like 0.5, so ETAA is bigger than ETAB. Okay, so I got another slide that gives you a very intuitive explanation of the situation. So this is my model of human Martian evolution. Let's say we are, on Earth, we are humans, but there are a bunch of Martians of a different species. But some of the Martians are sending migrants to the humans every generation. So the rate may not be that high. It's like less than one individual per generation. And then with the kind of population sizes are made up here, you could have this situation that we are all genetically Martian in the sense that let's say H1 is myself, H2 is you, and M is Martian. We get this probability that I join with a Martian first has a high probability than having you and me joining together first. Or in terms of the distance between two humans can be actually larger than between the human and the Martian. Okay, so that's really exciting. You can go and read the paper, look at the argument. So just talking with some colleagues who are working on species concept. Okay, I want to summarize. So the MSC is a very exciting, powerful, exciting framework for analysis of genomic sequence data. So right now we're just focused on closely related species. Full liking methods have some theoretical advantages. So they deal with the genealogical variations among loci. They deal with the phylogenetic uncertainties in the gene trees. If the species tree is hard, summary methods are far superior to the full liking methods are much better than summary methods. But if the tree is easy, all the methods will work. Okay, so some references, and then I just want to acknowledge the people who did the work. Thomas has been working on the BPP program, endless of debugging. Ziheng, I should mention, studied the impact of gene flow. Utaphan did the analysis of the allophoneous data. So thank you very much. welcome your questions.